Hi there, this is Dave Nguyen from the TSG Lab. The focus of my research is to develop algorithms to quantify shapes in 3 dimensions to understand biology. I've been working on methods to help crack the Glycan code, and in this presentation, I'm going to talk about one of them. Glycosylation is the addition of sugar molecules to proteins and lipids, and glycosylation is involved in almost every aspect of how the cell works, from protein stability to cell adhesion, all the way to signal transduction. We know that the structure of the glycan determines what happens to the protein and how the protein behaves. So we know that there is a glycan code, but we have yet to, quote unquote, crack that code. One of the reasons that it's been difficult to crack this code is because it is multidimensional as opposed to DNA. The code in DNA is linear, meaning it's a one-dimensional array of symbols, and it's semantic, meaning information is codified as a linear sequence of characters. The Glycan code, however, is nonlinear in that it consists of 3D branching array of symbols, and it's non-semantic, meaning information is codified in multiple dimensions. Therefore, in order to translate, like as in a language, a 3D code, we need a multidimensional approach. So here is the written description of the method, which I will not go through. The distance and connectivity matrix measures 4 features listed here for every single node in a glycan tree. Now, it's important to point out that the naming and ordering of the nodes does not matter, but what does matter is where you define the base node, because all distances are measured relative to the base node. This approach borrows from graph theory, which treats a tree as just a system of nodes and edges. So what you do is first, for every single node, you count the least number of steps to get to the base node. And you count along that same path how many nodes are first degree, second degree, third degree, and so on and so forth. So in graph theory, if a node only has one connection to another node, it's considered a first degree node. A node like E that has 2 connections is a second degree node, and a node like C that has 3 connections is a third degree node, so on and so forth. For example, if we were to count these measures for node F right here, we can see that node F is 5 steps away from the base node, 1, 2, 3, 4, 5. And the number of second degree nodes is 3. It's nodes E, D, and B. And the number of third degree nodes along this same path from F to A is 1. Only node C is a third degree node. You plug in all these 3 numbers to the node score equation. What you get is a value of 14. Now, the node score equation is summing 3 values. First is the number of steps to the base node. That is the least number of steps. Second is the number of second degree nodes multiplied by a weight factor of 2, representing second degree. And then third is the number of third degree nodes multiplied by a weight factor of 3. Now, if a tree has more than 3 degrees of nodes, then the weighting factor for each degree is the same as that degree, meaning if there's a 4th degree node, then the weighting factor is 4. This method creates a node score for every single node in a tree. Here is an example of a data matrix that results from applying the DCM approach to the structure shown in figure 3. Again, the naming ordering of nodes does not matter. What does matter is identifying the base node here, A. So if we calculate the distance and connectivity of every single node, what we get is the raw counts matrix. And then next, when we apply the weights for the second and third degree nodes, what we get is the weighted matrix. And on the weighted matrix, every single node has a node score, which was derived from this equation. So if you sum up this column of all node scores, what you get is one scalar value representing the tree in figure 3. And for this tree, the value is 61. And we refer to this as the distance and connectivity index, or the DC index. Thus, this method digitizes an analog glycan tree into a numerical matrix that can be compressed into one scalar value. But it digitizes it in a way that encompasses spatial information. In this case, its distance to the base node and the amount of connectivity within each node. In other words, we're not just translating an analog tree into text, which is just a re-representation of the information, we digitize in a way that encompasses spatial information. As proof of principle, we want to apply the DC index on a system of glycans that were related and that increased in complexity. Here's a diagram showing the synthesis of the core N-glycan structure, starting from only one monosaccharide all the way to its complete form in the endoplasmic reticulum. We calculated the DC index for every single structure along this path. When we graphed the DC index, we can see that there is a robust quantitative separation between the simplest form, which is only one monosaccharide, to the complete form. But moreover, if you notice the line that would connect these orange dots, this line isn't linear. You see that there's a shoulder here representing an increase in the slope of this line, and then the slope stays constant in this middle region and then increases yet again up here. This nonlinear line is very revealing, suggesting that the connectivity information we are measuring through this index changes in a nonlinear fashion, meaning the information in these molecules exhibits phases of growth and stasis. This kind of insight does not come from counting the number of molecules that are present, but is revealed when we measure the connectivity information in the molecules. That is the spatial information within the molecules. Next, we want to show that this DC index is able to quantify complex structures, both in normal development and in cancer. Cancer has been widely reported to alter the branching pattern of N-glycans, which dramatically affects cellular functions such as adhesion, migration, and metastasis. As you can see here, that an N-glycan modified in a cancer cell has a much larger amount of information or connectivity than do the typical complex glycans that exist in normal tissues. And here, there's only one difference in the addition of a fucose to the base node, and that results only in a difference of 1 unit. This tells us that the DC index weighs additions of monosaccharides at the distant ends of glycans more than it does addition of monosaccharides near the base. The DC index is precise enough to allow subtraction between two N-glycans, which is an index of how much information is different between them. Based on this precision at the level between two glycans, we hypothesize that the DC index will reveal insights at the big picture level between normal versus disease states. And it's important to point out that we are measuring complexity in the form of information, spatial information, not as enrichment for certain structures based on molecular counts. However, the two approaches will be combined. So what are the benefits of digitizing the glycan code? Well, firstly, precise measures along a gradient as opposed to categorical variables allows for better statistical correlations and machine learning predictions. Secondly, precise measurements allow for tracking of global ebbs and flows in biological information during normal development and disease. Third, an information-centric approach to measuring complexity extracts insights that counts of glycan molecules cannot. And lastly, our methods bring the field closer to cracking the glycan code. My overall hypothesis is that digitizing the language of analog glycan trees will allow for the extraction of quantitative themes in the flow, change, preferences, and bottlenecks of biological information in normal development and disease, analogous to how the quantitative study of human languages have revealed insights into how languages evolve and why. When you have a complex system of interacting parts, the rules of syntax create units of information like words in a language. Then the rules of grammar create denser structures of information like statements in a human language. These rules inherently result in preferences, which means there are mathematical themes that can be uncovered. This is true of human languages and will likely be true of the glycan code. For future directions, I'm developing several algorithms that integrate different aspects of spatial information in N-glycans, and eventually we will create an open source Python package, making the algorithms easy to use. We are seeking feedback to refine our approach, so if you have comments or questions, feel free to reach out to me at the email address on the screen. The rest of the document that goes with this video is supplemental data in which I show the calculations for each of the 15 structures of the N-glycan core structure so that people can verify the calculations and see that the system is simple enough that you can manually calculate it. Thank you for your attention. Please reach out if you're interested.